And welcome to this module's lecture of nucleic acid structure and DNA replication, chapter 11. I'm going to start off this lesson with a history of DNA. DNA isn't really that old compared to a lot of things that we know with science. How the biochemical identification of the genetic material happened. Okay, so there's 4 criteria necessary for it to be considered genetic material. That means it contains information, it can replicate, it has transmission, and variation. So in the late 1800s, biochemical basis of heredity postulated that research became convinced that chromosomes carry the genetic information. In the 1920s to the 1940s, scientists expected that protein, not DNA, but protein portion of chromosomes would turn out to be the genetic material. So we're starting off with this Griffith from 1928. This was the first major experiment that led to the discovery of DNA. Okay, so in the late 1920s, Frederick Griffin was working with Streptococcus pneumonia bacteria. So he had 2 strands of S. pneumonia bacteria. Strands that secrete capsules look smooth, and infections are fatal in mice. Strains that do not secrete these capsules look rough, and infections are not fatal to mice. The capsule itself shields the bacteria from the immune system, so they survive in the blood. So the smooth strands, called S, with capsules are fatal. The rough strands, called R, without capsules are not. So if a mice were injected with heat-killed type S, they survived because we killed the bacteria with the heat. However, mixing live R with heat-killed S kills the mouse. The blood is found to contain living type S bacteria. This is known as transformation. Diagram. If we look at A, okay, so the smooth strain, which is the disease causing, was injected into a mouse and the mouse dies. We have B, where the rough strain, which is not the disease causing, is injected into the mouse lives. We have C, we heat kill the S strain, the S strain is injected into the mouse lives. We heat kill the S strain, and D, we combine it with an R strain, we inject it together into the mouse, and the mouse dies. Okay, this is just another way to see that experiment. This is known as the transforming principle. So how was this possible that was happening? So the genetic material had been transferred from the heat-killed type S bacteria to the living type R bacteria. This gave them the capsule secreting trait and was passed on to their offspring. What was the biochemical basis of this transforming principle? We still didn't know whether it was DNA or protein. So at the time, there was no gave this Avery, McLeod, and McCartney, and they used purification methods to reveal that DNA was most likely the genetic material. So in the 1940s, there was an interest in finding the biochemical basis of bacterial transformation. They discovered only purified DNA from S type could transform type R. Okay, but purified DNA might still contain traces of contamination that may be the transforming principle. So that's why this wasn't the 100% definitive experiment. Okay, but they did add DNase, RNase, and protease. Okay, the RNase and protease had no effect. Okay, when the DNase was added, no transformation took place. It like inhibited it. And so this led to that it was DNA, but because of this contamination stuff, it wasn't 100% correct. The diagrams for you to read to see how that. Along came Hershey and Chase. Okay, these two used radioactive labeling to trace the DNA and the protein. They finalized and concluded that the viral DNA was injected into the cell and provided the genetic information needed to produce new viruses. Here we have the diagram of their experiment. Make sure to study it. And these are Hershey and Chase. So with that experiment, it was definitive that DNA was the genetic material. Then along came this Rosalind Franklin, okay, and she actually x-rayed the first photo of DNA with different techniques. She was important for that first photo, and then along came Watson and Crick. Okay, these had they said that DNA had specific pairing between nitrogen bases. DNA was made of two long strands of nucleotides arranged in a specific way called the complementary rule. And DNA that they described was the molecule from Franklin's X-ray. They said how adenine was paired with thymine and cytosine was paired with guanine. Okay, so these James Watson and Francis Crick put together these pieces of information, and they found this ball and stick model constituted with data. Oh wait, was consistent with the data. So they said it was a double-stranded helix, and they mentioned that base pairing with A and T and C and G. James and Crick and a guy named Morrissey Wilkins was awarded the Nobel Peace Prize for this in 1962. Okay, Rosalind Franklin had died, and the Nobel Prize is not awarded to people that have already died. And this was fine with Watson because it was always an argument between Watson and Rosalind because he hated the fact that Rosalind was a woman and didn't want her to receive any recognition for her contribution whatsoever. So next we're going to go over the actual structure of DNA. Let's start with nucleic acids. So terminology first, we have genes, which are units of genetic material that code for a specific trait. DNA is made up of repeating molecules called nucleotides. Okay, we have levels of DNA structure. Okay, we first have nucleotides, which are the building blocks of DNA and RNA. We have the strand, which is a linear polymer strand of DNA and RNA. We have the double helix, which are two strands together, which give it its shape. We then have chromosomes, and this is where DNA is associated with an array of different proteins into a complex structure. And then we have the entire genome, which is the complete complement of genetic material in an organism. Okay, so with the levels of DNA structure, okay, here we have a picture nice for you. We start with nucleotides to a single strand to that double-stranded helix. The DNA associates with proteins to form the chromosome, and that's what gives us our chromosomes. Okay, so we have formed from nucleotides these A, G, C's, and T's. So the nucleotides are composed of three components. We have a phosphate group, we have a pentose sugar, and we have a nitrogenous base. That pentose sugar is where the deoxyribose comes from, which gives us the D in DNA, deoxyribonucleic acid. The nitrogenous base is made up of two compounds, these purines and pyrimidines. The A and G are purines. The C and T are pyrimidines. Okay, so a DNA nucleotide. We first have that phosphate group. Okay, we have the sugar, which here we have pictured deoxyribose. And we have the nitrogenous base, which can be either A, G, C, or T. Okay, the diagram out of your book looks like this. It shows the phosphate group, the deoxyribose, and the base. Okay, this just happens to show A and G and T and C. With those nitrogenous bases, we have the purines as mentioned, which is A and G, and we have the pyrimidines, which are thymine and cytosine. Okay, and we have examples of what this may look like on the right-hand side. So dealing with the DNA strands, okay, what we can see here, we have that double helix. Okay, when we're talking about the DNA strands, okay, the steps of the ladder are the nitrogenous bases. Okay, so that's A, T, C, or G. So these here are either A, T, C, or G going across. The legs of the ladder, okay, are the phosphate and sugar backbone, pictured here on the side. So here we have that phosphate and sugar backbone making the outside legs, and we have either the A, C, T, or G making the inside steps. Okay, nucleotides are covalently bonded by a phosphodiester bond where the phosphate groups link the two sugars, and we have the backbone, which is formed from the phosphates and the sugars. The bases project away from the backbone, and it's directionally written from 5' to 3', which I'll be showing you here in a moment. So an example would be 5' TACG 3'. Okay, this is where we're discussing the backbone. Okay, the phosphodiester bonds. Okay, and the nucleotides inside. Now with that 5' and 3', we could see that here. Okay, in the this diagram. Okay, we'll see that on the next slide. So the structure was posed by Watson and Crick, where the DNA is double-stranded. We have anti-parallel strands. That means one goes up and one goes down. They are right-handed helixes. They have a sugar phosphate backbone. The bases are on the inside. The bases are stabilized by a hydrogen bond. Okay, so the backbone does covalent bonds. The in between does hydrogen bonds. There's a specific base pairing, and it says there's approximately 10 BP per helical turn. Okay, we then have Charcroft's rule, which states that adenine must pair with thymine and guanine must pair with cytosine. Their amounts in a given DNA molecule will be about the same. So with that double helix structure, we can see that here, okay? And we still see that 5', 3' end thing, which I'm going to relay to you here in a moment. But we know that it goes opposite because they're anti-parallel. So we have the 5', the 3' on this side, 5' on this side, 3' on this side. Okay, these are just base compositions of DNA for a variety of organisms. Okay, and so with those base pairings, it's also important to know they're base paired with hydrogen bonds. Okay, and G and C have 3 hydrogen bonds, and T and A contain 2 hydrogen bonds. So here we go with that orientation. So on the top rail, the strand is said to be oriented 5' to 3'. The strand on the bottom runs the opposite direction and is oriented 3' to. Okay, so I'm assuming nobody in here has had organic chemistry. Okay, but when we start pairing these molecules together, okay, we have like invisible legs of carbon coming off of these. Okay, there's 5 on this one and 3 on this one. This is where they get the 5' and 3'. Okay, so you just don't need to know how they get that with the organic chemistry, but just know that they run antiparallel as 5' and 3'. Now with the chromosome structure. So DNA coils around these things called histones to form a nucleosome, which coils to form a chromatin fiber. The chromatin fibers supercoil to form chromosomes that are visible in the metaphase stage of mitosis. Now, we haven't gone over the cell phases yet, but know soon we're going to be seeing one called metaphase. Okay, so as we can see in this diagram, we have the DNA tightly coiling together to go around these things called histones. Those histones coil around each other to make a nucleosome. They coil around each other to make this chromatin fiber. Okay, they get supercoiled. They then make chromatids. The chromatids are connected by the centromere. Okay, and we see it looks in an X form.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Irwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
Now that we've learned about the structure of DNA, we're going to learn about how it replicates. So an overview of that DNA replication. First, DNA replication. DNA is copied using original DNA strands as a template. In the late 1950s, 3 different models were proposed for DNA replication. They were called the semi-conservative, conservative, and dispersive models. Okay, so newly made strands are called daughter strands. The original strands are called parent or parental strands. Here we have an overview of the semi-conservative model. Okay, so as we're splitting up, we have the parent strand and the daughter strand. And this is the second round of replication. Okay, so the semi-conservative mechanism, DNA replication, produces DNA molecules with one parental strand and one newly made daughter strand. Mechanism, DNA replication produces one double helix with both parental strands and the other two with new daughter strands. First of model, okay, DNA replication produces DNA strands in which segments of new DNA are interdispersed with the parental DNA. Okay, you need to understand that we first have this S phase or interphase dealing with the cell cycle of the cell, which we'll be going over in the next module. Okay, but what we need to do with that is DNA replication occurs during the S phase or interphase of the cell cycle. Okay, so here we can see it in the green. This is the S phase. This is where that replication takes place. We will learn more about the cell cycle in the future unit. DNA first must be copied. The DNA molecule produces 2 identical new complementary strands following the rules of base pairing AT and GC. Each strand of the original DNA serves as a template for the new strand. Okay, so by the end, we have two. So the two parental strands separate and serve as template strands. New nucleotides must obey that ATCG rule. The end result is two new double helices with the same base sequence as the original. The replicated DNA molecules retain the same information as the original molecule. Watson and Crick showed the two strands of the parental molecule separate and each function as a template for the synthesis of a new complementary strand. Okay, as we've kind of already seen in the three models. Okay, and here we're showing the semi-conservative. Okay, we're showing the other ways of how they pair and separate. And we have these proteins for unwinding. Okay, so DNA helicase is an enzyme that binds to DNA and travels 5' to 3' using ATP to separate the strands and move the fork forward. Okay, it is responsible for unwinding and unzipping that double helix. We have DNA topo, I'm not sure how to pronounce that, so I'm sorry. This relives additional coiling ahead of the replication fork. And we have a single strand binding protein, which keeps parental strands open to act as the templates. We have RNA primase, which adds a short segment of RNA called an RNA primer on each DNA strand. Okay, so we have here a diagram showing what each one of those do. Okay, it'll make a lot more sense when I show you the videos at the end of this segment. Start off with DNA polymerase. These are covalently linked, this covalently links nucleotides, resembles a human hand with DNA threaded through it, and this deoxynucleoside triphosphates hydrogen bond to expose bases in the template strand. Okay, so here we can see it looks like a hand. Okay, and it pits together the template strand here, making a new daughter strand. The DNA polymerase cannot begin synthesis on a bare template strand. It requires a primer to get it started. So DNA primase makes the primer from RNA, and that RNA primer is removed and replaced with DNA later. DNA polymerase only works. Okay, so once again, we have it here in diagram form. When we show the video, it'll make much more sense. So we have a comparison of the leading and lagging strands. So what are leading and lagging strands first? First, we have the leading strand. DNA synthesized in as one long molecule. DNA primase makes a single RNA primer, and that DNA polymerase adds nucleotides in a 5' to 3 direction as it slides forward. The lagging strand is where DNA is synthesized 5' to 3', but as these things called Ozaki fragments. Ozaki fragments consist of RNA primers plus DNA. In both strands, RNA primers are removed by DNA polymerase and replaced with DNA, and DNA ligase joins adjacent DNA fragments. Once again, when you watch the video, this will make sense. Okay, so what's important to note when we're talking about this, when the DNA strand's open, okay, it's going to pair nucleotides on this side and on this side. Because they go in different directions, okay, it's a little tricky for the bottom strand. And that's where those Ozaki fragments come into place. And we'll show you that in the video. So proteins that are involved in DNA replication, okay, here we have listed their common name as well as their function.
DNA replication begins when the enzyme helicase unwinds a segment of the DNA and opens up the hydrogen bonding between the two complementary strands of DNA. DNA polymerase, the enzyme that synthesizes DNA, can add new nucleotides only to the free 3' end of a nucleotide strand. To provide a free 3' starting point, a short primer is synthesized first. Synthesis of the other strand of DNA is more complex. To provide a free 3' starting point on this strand, several short primers must be added. DNA polymerase then adds nucleotides to the 3' end of the primers. Finally, the enzyme, DNA ligase, forms a bond between the short DNA strands. During DNA replication, the leading and lagging strands are continuously synthesized.
The process of DNA replication in E. coli begins at a region of the bacterial chromosome called the origin of replication. The origin consists of an AT-rich region adjacent to sequences that are recognized by the DNA-binding protein DNA-A. The binding of DNA-A to the DNA-A boxes initiates a process that leads to an opening of the AT-rich region. The replication bubble in the DNA molecule is opened further by helicase, and the replication forks at each end of the bubble are expanded. Single-strand binding proteins then attach to each single-stranded DNA molecule to stabilize the open bubble, and subsequently topoisomerases bind to relieve the supercoiling of the double-stranded DNA created by the unwinding of the strands. RNA primers are synthesized by the enzyme called DNA primase. To make the two leading strands, single primers are synthesized at the origin. DNA polymerase III then extends each primer by synthesizing a daughter strand of DNA in the 5' to 3' direction as it moves toward the replication fork. Each lagging strand is made in the direction away from the replication fork. DNA primase synthesizes short RNA primers, and then DNA polymerase extends the primers by synthesizing a short segment of the daughter strand called an Okazaki fragment in the 5' to 3' direction until it reaches the RNA primer from the previously made Okazaki fragment. DNA polymerase I removes the primer and fills in the vacant region with DNA. Finally, two adjacent Okazaki fragments are ligated together by DNA ligase.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
So if you're still having trouble understanding DNA replication, okay, go and look at the videos that I posted for you in the video section, because it's really, really hard to grasp or understand this process without the visual video guiding you. Okay, so if you're still having trouble, definitely go and watch those videos that I have posted for you. So next we have these telomeres. These are a series of short nucleotide sequences repeated at the ends of chromosomes and eukaryotes. Now, if you notice with those videos, okay, one was a bacteria, one was a eukaryotic cell. Hopefully you caught that at the beginning of the videos. Okay, but these telomeres are specialized forms of DNA replication only in eukaryotes and the telomeres. Okay, so telomeres at 3' does not have a complementary strand and is called a 3' overhang. seen here. So DNA polymerase cannot copy the tip of the strand at the 3' end, so there's no place for upstream primer to be made. If this replication problem were not solved, linear chromosomes would become progressively shorter. So this telomerase enzyme attaches many copies of DNA's repeated sequence to the ends of chromosomes. Okay, so the function of this is associated with cancer. Okay, cancer cells divide uncontrollably. Okay, and 90% of all types of human cancers have high levels of these telomerases. Okay, okay, so here, okay, it goes through the whole diagram of we have one telomerase binds to a DNA repeat sequence. The telomerase synthesizes a 6 nucleotide repeat sequence. The telomerase moves 6 nucleotides to the right and begins to make another repeat. The primase makes an RNA primer near the end of the telomere, and the DNA polymerase synthesizes a complementary strand to the 5' to 3' direction. The RNA primer is eventually removed. Okay, and if this, continues, it can cause cancer.
The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from tetrahymena, which is a model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes the DNA strand complementary to the RNA found in telomerase, and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences, from a few dozen to several hundred. This prevents chromosome shortening.
Cells, like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, 2 identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
Okay, and that is the end of short, but there's a lot of information in it. I highly suggest that you watch the last video over again and you go into the video section and watch some of the YouTube videos that I posted to get a better sense of what the process actually is happening and with all the enzyme names and what they're actually doing. And as usual, if you have any questions, don't hesitate to ask.